So I'm gonna close here so I can have a little more of a and then I'm gonna come and share my screen. What happened? Sorry, my computer is not showing me the screen. There we are. I don't know what happened, but now I have 3 screens. Okay, can you see the presentation now? Now I'm trying to put you guys over here so I can see you. Perfect. Okay, so we were going into gastrulation. The previous slide was okay. So this is the last thing we saw last time. And we're looking at the primitive streak and how it places themselves on the posterior anterior side. And now we are going into how this streak allows and moves during gastrulation. Okay, so here is the posterior part of the embryo. This is the anterior, left, and right, and of course this is a dorsal view. So we talk about how the color cycle plays at the posterior end. It's like the embryonic shield that we saw in fish, right? So we have a thickening happening right here in this section, and is placed in the section of the epiblast. Remember, the epiblast is the section of the embryo that creates most of the embryo, okay? And this is where the color cycle is placed. So this is the section where gastrulation will start. So this thickening is caused by ingression. Remember the movement ingression, that going inside, moving in, okay? And it occurs during the ingression of the endoderm, okay? So from that epiblast. So they're going to start moving forward. They start going to migrate. Cells going to start moving here. This is all the Zona Pellucida, and this primitive streak going to start going forward, okay, towards the anterior part. Now, it moves very, very slowly, and as we're going to see in the next couple of slides, also going to have other type of movements. So migrate posterior to the anterior, traversing this area pellucida, okay, and this is the area opaca. Opaca is always on the outside, okay. This is something that you're going to be looking in the lab slides, okay. So this primitive streak defines the axis of the embryo. It extends from the posterior to the anterior section, and they were going to have a population of cells that are going to start gaining the inside of the embryo from the outside, from the dorsal side, everything that it was in that zona, okay? And start moving inside and moving forward on the embryo, Okay, so we want to see that a little bit. That line separates the left side from the right side of the embryo because it's going into the male sagittal section of that embryo. So the element close to the strake, after cell have passed through the strake, will be medial structure farther from the strake, and then want to go to the sides to construct this side structure. So let's see how this progresses. So here, we started here in the color cycle, and we're start moving in. And as we move forward, we have the creation of the primitive groove. Okay. This primitive groove is going to accomplish a very similar action as the gastrocele. Remember the gastrocele during the movement of gastrula in the amphibians, how it starts moving and creating that hole? Well, this is exactly what primitive groove is going to make. Okay, so we have the area pellucida that gets the hole in the middle that's primitive groove. And cells then start going from this area pellucida, the epiblast, into the inside of the embryo, gaining to the blastocele. Okay, so the primitive groove is the analogous to the amphibian blastopore. Okay, so at the anterior end, that primitive streak is a regional thickening. So in here we also have a little bit of a thickening where cells going to start gaining more the anterior portion of the embryo and becomes the Hensen's node. Okay, so in birds it's called the Hensen's node. As we're going to see later in mammals, it is called the node, just to make the difference. But the action is exactly the same. Okay, in mammals. So at the center of this node, you see in here, we're going to have a little bit of a funnel, and cells is a little depression, and that is called the primitive pit. So Hensen's node on the primitive groove, and at that end of the Hensen's node is the primitive pit. And through that, cells going to start passing through the blastocele. Okay. So Hensen's node is the functional equivalent to the dorsal lip of the blastopores that we saw in amphibians, and it's also the equivalent to the embryonic shell that we saw in fish, which is the organizer. So let's see how it goes. So as soon as the primitive streak has formed, we're going to have epiblast cells start migrating through and into that blastocele. So here we have that primitive groove moving forward. This is the Hensen's node up here. Okay, that's the Hensen's node, and we see how in the primitive pit right there, cells gain the inside of the embryo. So we're going to have three different populations of cells migrating in here. So we have cells that are start migrating through the Hensen's node at the first ones, the first group of cells, and will form the pharyngeal endoderm of the foregut. Okay, so this is they're going here and go right here into the anterior portion. Now, due to the different movements, these cells eventually will reach deeper parts of the embryo and displace the hypoblast, and eventually will migrate through the blood vessels and reach the gonad. So some of those cells will become the germ cells. Okay. Then the second group, this is what we have in here, the second group of cells entering through that Hensen node move anteriorly. As well, some will remain in the endoderm and the epiblast, and then they become the precordial plate mesoderm. Okay, so we're going to have these cells that are going to go also anteriorly, but some will go laterally, and that will become the precordial plate mesoderm. The third group of migrating cells passing through that Hensen's node become the chorda mesoderm. And we have already talked about the cord and mesoderm because we say that that will be originated in the notochord, right? So the cells that become the cord and mesoderm in here will subdivide into the head process. So we see that some still goes to the front, okay? That will differentiate into the forebrain and the midbrain. And then the other cells will become the notochord, okay? So we see as that primitive group projects anteriorly, everything that passes through that Hensen's node gives rise to the different population cells. So all of them, we see it in here, go to start migrating inside through that primitive streak, okay, going inside. And that's how we're going to have that plate mesodermal cells. And we're going to have at the end the epiblast, and then we're going to have the endoderm, and then we're going to have that precaudal plate mesoderm in this section. Is this image clear? Yeah, okay. So we have the Hensen's node with the pit, and then we're going to have the FEB as cells are going in. Those from the middle of the streak form the intermediate mesoderm and the lateral plate mesoderm. So we see it more or less in this section here. We're going to have that middle section and lateral plate mesoderm. For the posterior of the primitive streak, from the extra-embryonic mesoderm, that normally this is the one in pink color, okay? A little more pale, so those are the ones that are more towards the back, okay? We saw in Xenopus that some cells that pass over the blastopore lip in layers as they enter into the blastocoel, right? In chicks, cells entering the inside of the avian embryo ingresses as individual cells. So if I go back here, this is what we see here. These cells are ingressing. They are like delaminating, right? They are forming that intermediate mesoderm in that section. And the movement of the cells in the embryo normally is mediated by the activation of WNT in the epiblast, okay? And that is for those cells that are really close to the color cycle, so the ones that is originated here at the back, okay? As the primitive streaks are moving forward. The WNT pathway also activates the FGF. I think that by now you know that, right? And that's the fibroblast growth factor. And then we have the hypoblast. So we will see how the FGF and the WNT control this in a couple of slides. But for the moment, we're going to keep it there. So once the cells have moved forward and have gained the inside of the embryo, there is a nice phenomenon that occurs, and it's the regression of the Hensen's node of the primitive streak, okay? So that's a new phase of gastrulation. The first one is the epiblast cells gain the inside of the embryo through the groove of the primitive streak and the Hensen's node. And once they have reached and gained the section of the pharyngeal, that Hensen's node starts regressing, which means moving back. Remember, we have a groove, we have a hole, right? So the Hensen's node going to start regressing to almost close the Velcro, okay? Like a zipper going down and closing that groove. So the Hensen's node start moving towards the center of the area pellucida to be more posterior position, okay? It leaves in this wake the dorsal axis of the embryo and the notochord, so two structures that can only be formed once the Hensen's node start regressing, okay? At this time, we're going to have all the presumptive endodermal and mesodermal cells have entered the embryo through that pit. The epiblast is composed entirely of presumptive ectoderm now. Excuse me. And hence, we have that the sequence in which the head, mesoderm, and the notochord are established in the avians, in birds, and in mammals, we want to have a distinct gradient that is anterior and posterior. The parts that are anterior are more mature than the parts that are posteriorly, because we need that Hensen's node to regress. Okay? So we're going to have some cells still going through gastrulation at the back, but we have cells doing neurulation at the front of that embryo. Is this concept clear? Yep. Okay. So two different tempos. Okay. Gastrulation is still at the back, but neurulation happening already in the anterior portion. So for the next several days of development, the anterior end of the embryo is more advanced in its development, and that is a head start for that embryo. Okay. So this is something that is really important, and we want to read a paper on this. I'm going to put it on perusal. This is for after the break, okay? Because I said that for this Friday you will not have a paper to read, so that will be for when we come back from the break. So now we have that section going to the front, and now we have the Hensen node coming to the back, and now we have this anterior-posterior axis, right? Because we, as the Hensen's node regressing, the parts of the anterior section are more developed. So there is a difference in how the anterior-posterior axes are established for the mesoderm and ectoderm point of view. The ectoderm, most of the initial neural plate corresponds to the future head region. We're going to have a small area of the ectoderm that is called the caudal lateral epiblast. This is the caudal lateral epiblast. So it's on the side, as we see here. Okay. This is the notochord section. And this caudal lateral epiblast will form the nervous system, including the posterior hindbrain, as well as the spinal cord. While the Hensen's node is still regressing, because this is the Hensen's node that is still regressing here, okay, we're going to have a huge amount of the FGF, the fibroblast growth factor, that gives the signal that the straight and paraxial mesoderm, which will be the future somite, do not differentiate, which could be considered by deduction as an antagonist of the retinoic acid that we have seen before, right? We need some inhibition for that section, okay? And that's what we saw in fish. Now, for the mesoderm, it receives instructions that tell them exactly where they are along this anterior-posterior axis. And the entire length of the notochord at this midline here is derived from cells that are present in the Hensen's node, okay? So this one here doesn't change, and that's for the mesoderm. So we can have cells still going through that Hensen's node as it regresses, okay? So the anterior-posterior identities along this rostrocaudal axis from the hindbrain to the tail are specified as a function of the, be careful here, time. Remember that I told you a couple of weeks ago that time was very important here, okay? So this anterior-posterior depends on the time of the emergence of the primitive straight and the Hensen's node regression. So let's see how this works. So in this figure, we observe the signals that regulate the axis extension in the sheep embryo. So we're just only gonna follow the numbers here, okay? And these arrows. So first, we see that FGF 8, that is here with the number 1, inhibits the expression of retinoic acid. Okay, so that's the RA. So we have a first inhibition of FGF and retinoic acid that normally synthesizing the RALDH2, this one that we have in here. So we have here FGF that's gonna cause an inhibition of this one that is normally the one that is in charge of the retinoic acid. This occurs in the presomatic mesoderm. Okay, all of this is the presomatic mesoderm. Okay. We also have the expression of retinoic acid receptor, the RAR beta. Okay, so we have this here. Okay, in the neural ectoderm. We know that is the neural ectoderm because it's really close here to the middle line. Now, these 2 steps prevent retinoic acid from entering differentiation in the caudal. So we have all of this purple color. We know that is FGF, and we are inhibiting the expression of retinoic acid in that caudal lateral epiblast cells. Okay. Now, FGF is also inhibiting a friend of us, the Sony hedgehog, SHH. Okay. So it's inhibiting the expression of this in the neural tube floor plate. Okay, and this is controlling the onset of the ventral pattern. So inhibition of retinoic acid, but at the same time, inhibition of Sony hedgehog. FGF signal is also required for the expression of delta, the delta 1 in the middle portion of the caudal lateral plate. But this one also promotes the expression of WNT8C. So FGF really important to determine that caudal fate in here. So to maintain this group a little less, susceptible to the effects of retinoic acid, which is very well expressed in this section as the Hensel's node regresses. Okay. So there is a point when FGF start decaying in the caudal paraxial mesoderm. And then WNT8, which is a little farther towards the middle of the embryo, becomes the source of signaling that promotes this RALDH2 in the paraxial mesoderm, and also it promotes the retinoic acid produced by RAL2 to express FGF. So then we have a positive feedback. Does this figure make sense in here? So what we want specifically is this FGF to exert an inhibition of the retinoic acid. Okay. Now, the conversion of the radially symmetrical blastoderm that we had in here, right, to a bilateral symmetrical structure with an anterior-posterior polarity is accomplished simply by gravity. Okay, so we have here the egg in the oviduct. Okay, and this egg normally do rotations very slow. Normally it takes to complete a full rotation is about 20 hours in the shell gland. So just to have the shell produced. So it goes very, very slow because it's rotating. In here, we're going to have about 15 revolutions per hour. Okay. And what happened when we put things into a centrifuge, right? All the components are light, gonna lie beneath one side of the blastoderm. So this is our blastoderm, and everything that is light gonna go this way, and everything that becomes heavy gonna become in this way. So this is where the rotation comes in here. So this step up, normally the one that we have in here, of the blastoderm is elevated and becomes the posterior portion. So everything that is like comes the posterior portion of the embryo where the primitive streak forms at the beginning. Okay, and the portion of the blastoderm becomes something that we're going to be calling, we're going to see that later, the posterior marginal zone. Okay, so it contains all the cells that are the equivalent to the nucleus center. That posterior marginal zone produces WNT8C, the one that we saw in here, that is causing the inhibition of the retinoic acid later, as we see here. Okay. And it's capable of inducing the accumulation of beta-catenin. Beta-catenin, once again, this molecule, paracrine factor in here. And that zone also going to have the production of VEC1, another factor that we have seen before, that is also produced part of the TGF-beta superfamily. Together, WNTC and VEC1 induce the expression of nodal. Nodal is another old friend that we have, right? Member of the TGF-beta superfamily in this embryonic epiblast in here. So next to the color cycle. So let's see here with a little bit more of the names. So this is in frog, this is how we saw it, and this is in the chick. Okay, so we have beta-catenin in the area of PECA, right? All the way around here. We have VEC1 expression in the posterior end. And then we have the nucleus center is going to be formed at the junction of the two of them. So in this section here, okay? So nucleus center happens exactly the same way as we saw in frogs, but now in chicks. Now we have left this left and right axis, right? We know how the anterior posterior was formed and the dorsoventral. The distinction between the right and left sides in vertebrate also, once again, is primarily controlled or regulated by two proteins, the paracrine factor nodal, right, and the transcription factor PTX2. As the primitive streak reaches its maximum length, so all the way up here, okay, Sony hedgehog, which is a transcription factor gene, becomes restricted to the left side of the embryo, which is controlled by activin. Activin normally induce Cerberus here to induce the BMP to produce nodal and activate PTX2. So here we have that in the right side. So we have Sony hedgehog, which induces Cerberus, and their Cerberus induces BMP, and PMP induces nodal, and nodal induces PTX2. This is all in the left side. In the right side of the embryo, activin and BMP, the BMP here is BMP4, okay, blocks the expression of Sony hedgehog, but not necessarily the expression of FGF8. FGF8 then blocks the expression of Cerberus, and if there is no Cerberus in here, there is no activation of nodal or PTX2. Are we clear in this image? How we determine the left and right axis? And at the end of that, if things goes well, we have a little chicken or a little bird of form. Now, in mammals, we know that cleavage has been the most difficult to study because it's in utero. Okay. So mammal eggs are among the smallest in the animal kingdom too, which makes things a little harder to manipulate experimentally. Mammals don't produce a heck of a lot of eggs either, which makes things a little also more complicated in this regard. Like we saw in sea urchin, fish, and amphibians, there is mostly one at a time that is produced. And of course, mammal embryos develop inside, so it's hard to see that development. Although there are new studies that are becoming a little better and creating more of an artificial placenta. Okay, so mammalian cleavage is very different. Have 5 unique features. Fertilization occurs in the oviduct en route to the uterus. First cleavages occur during that journey, so it's way harder to see them occurring. We have a rotational cleavage, very similar to what we saw in birds. The asynchrony of the early cleavages is different because in the other animals we saw that the first cleavages have a very organized way to present, right? The genomes are activated early and they go through a process that we call compaction. So let's see these steps. So this is the development of the human embryo from fertilization to the section of implantation in here. So this is the ovary. We have the Greffian follicle that has released the ovule during the period of ovulation. So the fiambare catches that egg. And then here in the section that we call the ampulla, that is all this section here, fertilization normally occurs. If fertilization occurs right here, we're most likely going to have an ectopic implantation, which normally is doesn't get to fruition, right? So normally during the ampulla section here is where fertilization should occur, okay? Fertilization then gonna triggers the completion of meiosis, as we have seen last week and in the lab, okay? The first cleavage begins normally 24 hours after fertilization, so the process is a little longer. We know that we have an egg that have a very thick layer surrounding them, okay? So first section occurs 24 hours after fertilization, and cleavage in mammal embryos are among the slowest of the animal kingdom. Normally between the first and second cleavage, we can have 12 to 24 hours apart, while the other ones happens very, very quickly, okay? So as they start progressing with the help of the cilia of the oviduct, the embryo gets moved all along the fallopian tubes until it reaches the uterus. So when it gets into the uterus, it should be in the state of blastocyst, okay? Almost ready to hatch and be able to proceed with the next step, that is the implantation in the wall of the uterus. In terms of division, we see that they have a rotational cleavage, where the first, here we have the panel of the first cleavage is meridional, okay? However, the second cleavage, one of them blastomeres divides meridionally, and the second is equatorially. So here we have first plane, second plane, meridional. This is in amphibians and echinoderma. But in mammals, we have one that is meridional, but the second is equatorial. And that is why it becomes rotational, right? These ones were meridional, but in this one is meridional. And then one is meridional, but the second is equatorial. The other thing that I say is that it has a marked asynchrony in the early cleavages. So mammalian blastoderm do not all divide at the same time, okay? Mammalian embryo often have odd numbers of cells rather than, as we saw in the other species, 2, 4, 8, 16, 32. We're going to have an odd number in here. Then we have the genome are activated early. So unlike almost all other animals' genome, mammalian genomes are activated very early. And this is to be able to produce proteins that are necessary for cleavage. In mice and goats, the switch from the maternal zygotic control occurs normally at the stage of 2 cells. So here we want to completely change the whole thing. The last step is compaction, and this is unique for mammals. And we see this in these mouse blastomeres that are not going from the 2nd up to the 8th stage cells that form mainly a loose arrange of cells, right? We see here 2, then 4, and then here 8. Still, the cells look very loose in here. Once we get into the 3rd cleavage, here, we want to start observing a spectacular behavior of these blastoderms. The cells suddenly start huddling together to form a compact ball of cells. In order to do that, they use a specific type of molecule that we have seen before that helps for the addition. What is the name of that molecule that allows cell addition in embryo? Don't remember? Cadherins, okay? So the E, and for the epithelial cadherins, remember there are 5 different classes of cadherins, okay? In this case, it's the E cadherins that are expressed in the blastomeres, that are expressed in the blastomeres and allow them to take something that is very loose into something that is very compact. And we see it here, right? It's more like a ball. So this tightly packed arrangement is normally stabilized by tight junctions that form between the peripheral cells. And this is where we see a little bit more here in F, right? So comes from a sphere point of view. The cells within the sphere form gap junction, and we're going to start stabilizing with different type of molecules. The cells of the compacted 8 embryo normally produce a 16 embryo that we call the morula. So if I may come back to here, so we have 2, 4, 8, 16, and we are in a morula stage. And we see how it becomes loose to a compact ball. Okay. So the morula consists now of 2 groups of cells. We have internal cells and we have external cells. And here we go again. So in this is our morula with the internal cells, which we're going to call the inner cell mass in this section, okay? And we see that most of the adjacent to the external cells become what we call the trophoblast or the trophectoderm. That's a word that every time I have, I need to think twice before I say it, okay? So we have the trophoblast on the outside, that is the trophectoderm. And then we have the inner cell mass, or ICM, okay? This inner cell mass gives rise to the embryo, not the external, not the trophoblast, but the inner cell mass. The embryo will rise from this, okay? And it normally becomes positioned on one side of the ring. So this looks like a ring, right? So it's gonna be in this section. So this is the inner cell mass that gives rise most to the embryo. And then we have the blastocyst. So we have these changes from morula to blastocyst with the blastocele in the middle, the inner cell mass on one end, and then all the external cells are the trophoblast. Okay, so here we get the loose cell, and then it's here when it goes into compaction. Okay, so these trophoblast cells, what they do produce, this will produce no embryonic structures such as the chorion, the extra-embryonic membrane, and a portion of the placenta. Okay, so the placenta is really important because that enables the fetus to get oxygen and nourishment from the mother. So they can become vampires somehow. Okay, so the chorion, it plays a very important role because the chorion also secretes a set of hormones forcing the mother not to reject the embryo by regulating the immune system of the mother. Remember, that fetus has half of information that is from the father. So the mother immune system will say, Houston, we have a problem. There is an strange, there is an alien in here, right? So the chorion gonna play that role to inhibit and to force the immune system of the mother not to reject this information. So this, yes?
I have a question. So when there is a rhesus conflict between the fetus and the mother, it's known that the second pregnancy with the rhesus conflict is more likely to get rejected. Is it because mother's body is already adapted to a similar set of hormones?
No, the rhesus is only the rhesus D. In the second pregnancy, it's only if the mother gets a little tear somewhere, that normally tears happen during normal delivery, right? And then the blood of the mom picks up the signal that the blood of the baby was not compatible with hers. So in the first pregnancy, nothing happens, right? It's only in the second pregnancy. And that's what normally we want to have the vaccine anti-D given to the mother after the first pregnancy to avoid a refusal early on.
Okay, thank you.
That makes clear, Ilana?
Oh yeah.
Okay, perfect. So that chorion going to play a very important role to avoid the rejection because the immune system of the mother is going to detect. So the early cells that are placed in the trophoblast are, say, totipotent. Okay, so they can become any type of cell. They can produce the inner cell mass and the trophoblast. The early cells from the inner cell mass are pluripotent. Okay, so they can become many type of cells, but not all of them. Okay, so they can produce any type of cell of the body except the trophoblast. Okay. The mammalian embryo proper is derived from the descendant of the inner cell in that 16-stage morula. And we're going to have some supplemented by cells that are derived from the trophoblast, so that Ectoderm. The cells that generate the inner cell mass give rise to the embryo and the extraembryonic membranes, that is the yolk sac, the allantois, and the amnion, but not the chorion. The chorion is from the trophoblast, okay? Once that embryo has reached the 64 cell stage, normally we're going to have that the trophoblast becomes separate cell layer, neither contributing to the cell or any other group. The distinction between this inner cell made up of pluripotent cells are, as I said before, And trophoblast is the first differentiation in mammalian development. We haven't seen this in birds, right? So this is what separates them really well, the distinction between the trophoblast and the inner cell mass. And this differentiation, as I explained it a couple of slides ago, is really important because we need to do the implantation of the embryo in the endometrium wall of the uterus. So the development of the embryo proper can take place only if it attaches to that wall of the uterus. So after the morula, we saw that the morula is going out of the fallopian tube, right? Getting at the onset of the uterus, okay? So after the morula forms, cavitation occurs. And in here, what we have is that the cells of the trophoblast start secreting a fluid into the morula to create the blastocele. So I'm just going back here. So we are here at the onset, okay? And cells start secreting to have the blastocele in here, okay? So that blastocele is created, and they do this by using some sodium-potassium ATPase and sodium-hydrogen pumps. So they are using two different types of pumps, sodium-potassium and sodium-hydrogen pumps. And all of that is coming from the trophoblast, okay? And by using those pumps, they are creating an osmotic gradient that is drawing water in to create that blastocele. So the resulting structure is the blastocyst, is yet another structure unique to mammal embryogenesis. We haven't seen that in any of the other groups. The next step that is required is to escape from the zona pellucida, okay? So while the embryo moves through the oviduct towards the uterus, the blastocyst starts expanding with the zona pellucida. The zona pellucida at the same time prevents the blastocyst from adhering to the walls of the oviduct. So it's protecting them. And by that, we avoid the ectopic pregnancy in most of the cases. So that's what I say, that if the egg is fertilized at the very end or entrance, more or less, of the fallopian tubes, it will hatch before it gets into the uterus. Okay, so that zona pellucida is protected there. So here we have, and here we have our blastocyst hatching out. So when the embryo reaches the uterus, it must hatch from that zona pellucida that we see in here. Okay, and this is made through the secretion of strepsin. It's a protease on the trophoblast cell membrane that lies as a hole into the zona pellucida, and the blastocyst starts squeezing out through that zone. Once it's outside, and this is what we have here, some that has hatched, okay, they can make direct contact with the uterine wall. And this is what here we have. This is the endometrium of the uterus, and this is the hatch blastocyst. This is the trophoblast, and here we have the inner cell mass. Now, how they attach? I have heard stories of females that know exactly when this happened because they say that it hurt. I didn't felt anything myself with my 2 pregnancies. So in here, we have the uterine wall, okay, the endometrium, that almost like catches the blastocele on its extracellular matrix. Now, this trophoblast all around contains some integrins. Remember, integrins are molecules for attachment, right? We saw them way at the beginning of the semester. So these integrins normally gonna bind to the uterine collagen made with fibronectin and laminin, two molecules that we have seen in the past that works for addition of cells, okay? But there is also a special cadherin, and it's the P cadherin that is for placenta, P for placenta cadherins. And that one is present in the trophoblast and also in the uterus endometrium. So both ways, these ones allow to catch that blastocyst, okay? Now, once in contact with the endometrium, the trophoblast encourages normally the expression of WNT. WNT encourages the cells to secrete another set of proteases. Those proteases, what it's going to do is going to dig a hole in the extracellular matrix of the uterus, okay? And enabling the blastocyst to bury himself into the uterine wall. And this is the part that some females have said that they have felt when the blastocyst opens that hole in the uterus wall to niche in the wall. And it seems very dramatic. So here we have all the different steps that the blastocyst takes. So day 5, normally that blastocyst gets into the uterus. So this is 5 days post-fertilization, okay? So the inner cell mass, that is what we have right here at the bottom, and delimitates the hypoblast here in green, okay? And then here we have the blastocyst, okay? And then we're gonna start having the formation of the extraembryonic endoderm. And this is what will become, the primitive yolk sac and the bilayer epiblast and hypoblast that will become the blastodisc. So this, I'm just trying to change here. So here we have the trophoblast all around. This is the epiblast, and this is the hypoblast, and this will become the blastodisc in December. Okay. So, and then a couple of days pass. This here is where we have the connections with the cadherins and WNTs and all that. So we just start attaching them. By day 8 and 9, we see how we are opening that hole in the wall of the uterus, okay? By day 10 to 12, the trophoblasts start changing, okay? So here are two important elements. We have the syncytial trophoblast, okay, that ingresses into the uterus tissue to form what will become the chorion. So that's the syncytial trophoblast, okay? And this comes from the villi of the egg. And then later on, we see how it is completely immersed. It is important to open that hole to make bleed that uterus cavity to establish by angiogenesis all the blood work between the embryo and the mother, okay? If it doesn't bleed, you cannot attach to the surface. You gotta establish that connection with the blood system of the mother. So here we have the trophoblast that was happening here, divides into this cytotrophoblast, okay? That is the one that is surrounding, and the syncytial trophoblast that ingresses and allow the embryo to niche inside. Day 15, or 12 to 15, I should say, gastrulation will take place, and this is what we have seen in here with the node, that this is the primitive streak, okay? The epiblast is split into the amniotic ectoderm, okay, which circle the amniotic cavity. We're gonna see it in a different slide, Okay, and the embryonic epiblast. So if we have here our first group, okay, so we have the trophoblast, cytotrophoblast, and the syncytial trophoblast, which is this one here. This is the blastocyst. So we have the inner cell mass, that is the group that's going to form the embryo. So we're going to have the epiblast, which is this section here. Okay. That epiblast will become the embryonic epiblast, and we're going to have the 3 germ layers from that primitive strake. And we're going to have some of the extra embryonic elements, not all of them. In the other hand, we have also some of these membranes that are going to be forming, as we explained before. Gastrulation, how this gastrulation occurs. So as bird and mammals are both descended from reptiles, we know that mammalian development has some similarities to reptiles and birds. Interestingly, the gastrulation movements of reptiles and birds, which evolved to manage development with an enormous amount of yolk, are still present in mammals, even though we know that the placental mammals have isolated eggs, sparse, evenly distributed yolk. Okay, they keep some similar steps. So here we have the amnion structure in human, as we see it is happening. So we have our, in here, we have the extraembryonic mesoderm. This is the syncytial trophoblast that allows the insertion of that embryo. We have here the amniotic cavity. Here we have the bilaminar germ disc with the epiblast in gray and the hypoblast in green. And then here we have the yolk sac. Okay. So the first segregation of cells within the inner cell mass results in the formation of these 2 layers. So before we only have 1 layer, and then divides, and we have these bilaminar layers: hypoblast, which is the primitive endoderm, and the upper layer that is homologous to the hypoblast, which is the epiblast. Okay, the epiblast and the hypoblast form this bilaminar germ disc. The primitive strake endoderm cells get in contact with the epiblast in the visceral endoderm. The yolk sac, which is this portion over here, get in contact with the trophoblast, that is the one that was surrounding, and become the parietal endoderm. This epiblast cells layer splits, and then we're going to have the formation of the amnion, which is what we have in this section over here. Once the formation of this amnion is made, it fills with the amniotic fluid, which normally, as we say, allows for the shock absorption of the embryo and also prevents the desiccation of the embryo. This embryonic epiblast is believed to contain all of the cells which form the actual embryo, and thus is very similar to the avian epiblast, as we saw it before. As we saw in chicks, gastrulation begins in mammals in the posterior end of the epiblast, where the Hensen's node forms. But as I say, to differentiate it from the birds, we just call it the node, the Hensen's node. But the role is exactly the same. So this is the amniotic cavity. And here we have the bilayer embryo, epiblast and hypoblast. Okay, and we have the posterior and anterior side of the embryo. Okay, so the primitive strake is the one that is forming here. Okay, and cells start migrating from the node to form the notochord. Okay, Now, there is one difference here, and it occurs with the chorda mesoderm. The chorda mesoderm cells, which want to form the notochords we have seen prior, can be seen as a band of small ciliated cells between large endodermal cells and extending rostrally to form the node. So these are the ones, these arrows going up here. So this is a 14 to 15 day. We have the primitive groove, we have the node at the end, and we see how cells start gaining the inside of the embryo through that groove and the node. Okay. Cells migrating and the specification of the cells normally is coordinated by FGFs, so by fibroblast growth factors. And these factors are coming from the primitive straight, the same thing as we saw in chicks. Okay, so FGF, once again, control the cell movement into that primitive straight by downregulating E-cadherins. Okay, we know that these cadherins were used to compact those cells, remember? So now FGAs will downregulate that E-cadherin that holds the epiblast together and start controlling now these E-cadherins, these nail, brachyury, and TBX6 genes. The same type of genes that we saw in chicks. Now, how we form those extra embryonic membranes? So being viviparous requires a dramatic structuring of maternal anatomy, which means also the expansion of a portion of the oviduct into the uterus. The remodeling requires the development of a fetal organ capable of absorbing the maternal nutrients, so they become really vampires. And we say that that structure was the chorion. Okay, so chorion derives primarily, what can I say it? From cells from the trophoblast, that's what we said, and the chorions from the fetal portion of the placenta. So we have two structures. We have a placenta that is from the embryo, and another side of the placenta that is made from the mother. Okay. So the chorion enables the fetus to receive oxygen and nourishment from the mom, but also secretes the hormones that block menstruation. If the lining of the uterus is allowed to shed, bye-bye fetus, right? Because it's sticking into it. So it produces regulators also of the immune response from the mom, so the embryo is not rejected as a foreign tissue. So chorion induces the endometrium to form the maternal portion of the placenta, which we call the decidua. So here we have, this is our embryo, the umbilical cord, this is the amniotic sac, and here is the side of the embryo, but on the other end, stick to the wall of the uterus, it is the placental side, that is the decidua. So the decidua is well vascularized and will provide oxygen and nutriments, and we see it here very, very well vascularized to provide everything that embryo requires to grow. So how we do this? So the initial trophoblastic cells undergo normal divisions, but later they undergo mitosis without cytokinesis. Remember, cytokinesis is the splitting of the cells. So they start increasing the number of cells, but without dividing, without separating from one another. And that's how we get two tissues formed. The cytotrophoblast, which initially adheres to the endometrium through the addition molecules using the p-cadherins, okay? This cytotrophoblast secretes paracrine factors that attract the maternal blood vessels. And with those proteases, okay, also will remodulate the uterine wall and blood vessels so that the maternal blood bathes fetal blood vessels. As I say, if it doesn't bleed, they don't establish connection. Okay. The second layer that we have is the syncytial trophoblast. This syncytial trophoblast digests the uterine tissue to allow the embryo to embed deeper into the uterine wall. So that was what we saw in the gray color, little gray pinky. Where is it? Here. Okay, so this is our syncytial trophoblast that will just really dig down the wall, digest that wall to allow the embryo to embed in the wall. So the uterus responds by increasing the vascularization in the adjacent tissue. So this is all about this healing process, right? When we break up a blood vessel, angiogenesis, new creation of blood vessels need to occur. And this is how they do it. So that little embryo, vampire, hurts the mom in order to be able to grow inside. Okay. So these new vessels contact the syncytial trophoblast and eventually contribute to the formation of the blood vessels of the umbilical cord. Is this clear? Yeah. Okay. So this is a 615 week embryo. So it's about 50 days, right? And it's a human embryo. Okay, just to show this extraembryonic membrane. So the embryo is encased in the amnion. So this is the embryo, and he is very cozy in that amnion, and it's further shelled by our chorion. So here we have our chorion all around. So he is really well encased in this section. We see a lot of blood vessels extending to and from that chorion, right? Around here. And then here we see the villi of that chorion projecting from outer surface of that chorion. So we do have some of analysis that is done with the villi. We're going to be talking about it because this has the information of the baby, okay? And then we have this little sphere over here that is the yolk sac. So very tiny in that section. That doesn't feed that baby for too long. Okay, so this anterior-posterior axis. For this, we have two signaling centers. The extraembryonic tissue is very critical in the polarizing the mammalian embryo and to forming its two major signaling centers. One is the node, which is the organizer, right? And the second one is the anterior visceral endoderm, or AVE. So the node is responsible for the creation of all the body, responsible for the neural induction, and for the pattern of most of the anterior-posterior axis. Very similar of what we saw in the chicks, right? So many tasks. On the other hand, the anterior visceral endoderm has some overlapping functions, but its main function is for the positioning of the primitive streak. So the two centers work together to create the forebrain. That's what they say they have some very similar functions, overlapping functions. So the signal that initiates the primitive streak comes from the interaction between the trophoblast derived extraembryonic ectoderm and the epiblast. So if here we have our embryo, this is the amnion, this is the allantois. Here we have the embryonic ectoderm with the amniotic cavity, and here we have the anterior visceral endoderm. Okay, and in red we have the precaudal mesoderm. So BMP4, and we're going to see this in the next slide a little better. Okay, BMP4, we're going to start with that one. From the extra embryonic ectoderm and induce the epiblast to make WNT3 and nodal. All of that is BMP4. AVE, in the other hand, prevents WNT3 and nodal from having an effect on the anterior part of the embryo. And it does this by secreting some paracrine factors, LEFT1 and Cerverus. Once formed, the node will secrete cording and noggin. And this is the same as we saw that was expressed in the organizer tissue of the frog. So let's see in this figure. So this is the posterior part, this is the anterior part, this is dorsal, and this is ventral. Okay? So the head region normally is devoid of nodal signaling. So here is the BMP, FGF, and WNT that we know there are nodal. Graded gradient in the posterior, almost nothing in the anterior. This is where we have some antagonists here in the front. So this posterior is really characterized by nodal, BMP, FGF, WNT, and also a molecule that we have seen in the past, that is the retinoic acid, right? Retinoic acid normally is placed at the posterior end. So here, what we have is that graded gradient of WMP, WNT, BMP, and FGF highest at the posterior than at the anterior part of the embryo. And this is what this primature streak and other posterior tissues are the source for these WNT and BMP proteins. So the organizers and its derivatives, the notochord, produce the antagonist in the anterior portion of the embryo. The cascade in here, we see normally that the FGF in the late gastrula has a gradient of increased retinoic acid, with retinoic acid levels being high in the posterior region. Then we can control different enzymes and genes that will activate the specific genes. So FGF8, WNT3A, and retinoic acid contribute all to that posterior embryo working together with some related genes. There is the CDX1, CDX2, and 4. And all of these control the Hox genes, get activated. And I'm just seeing that it is 11:17. And, oh, come on. I just want to get back to my screen. Come on. So we're going to leave there for today. And we will resume on Thursday to finish up. We are on slide 38 and we have 50. So we are going to finish this and start our next subject. Okay. I hope that you all have a good week, a productive one. If you have some midterms or things like that, I wish you the best on those exams this week. And just breathe. Everything's going to be okay. Okay. See you on Thursday.
Have a good day.
Bye.
Thank you. Have a good week.
You too.
Have a great day.
Thank you. Well.
Thank you, Professor.